This is a mitotic phase or the phase of equational division as the cell undergoes a complete reorganization to give birth to a progeny that has the same number of chromosomes as the parent. The other organelles are also divided equally by the process of cytokinosis, which is preceded by mitotic nuclear division. Okay, after nuclear division, cytokinosis happens, which we will discuss later. So why is it called equational division? Because there is equal division of everything between the two daughter cells. Okay. The mitotic phase is divided into 4 overlapping stages: prophase, metaphase, anaphase, telophase. These are the 4 phases that are included in the M phase. Okay. So this is what mitosis looks like. This is the prophase, metaphase, anaphase, and telophase. So let's discuss the prophase. Okay, we'll keep coming back to the diagrams as well. Prophase. The completion of prophase is characterized by the initiation of the assembly of mitotic spindles. Okay, it is not clearly visible in this one, so I will go to the other one. These are mitotic spindles. It is so this phase is basically characterized by the presence of mitotic spindles, and the centrioles go to the opposite poles. Okay, these centrioles, this is the acting force. Okay, the nuclear envelope disappears. Okay, let's see. The microtubules and the proteinaceous components of the cytoplasm that helps in this process. So what are mitotic spindles? These are basically proteinaceous components. Okay, these are the things holding the nuclear material, the chromosome over here, because the nuclear membrane is going to disintegrate in this process. Okay, the nuclear envelope starts disintegrating. The microtubules are allowed to extend from the centromere to the chromosome. Okay, from the centromere to the chromosome. These are the things holding up. Okay, this is the prominent machinery over here. The other organelles. Okay, so where were we? Okay, the microtubules attach to the kinetophores, which allow the cell to move the chromosome around. So what are kinetophores? Kinetophores you see over here, centromere. Okay, just adjacent to the centromere, there are kinetophores that help the chromosome to get attached to the microtubules. Okay. Okay. Now let's do a question and answer. Quick question and answer. Individual chromosomes become distinct through a light microscope during this mitotic stage, which is obviously, yeah. Okay. So during prophase, one more thing, the chromosome become very condensed. Okay, very condensed. They will become very condensed. That is the reason you will be able to see them through light microscope. If you see it under the microscope, the prophase will be visible very prominently. And what will be happening? What will be happening is that you can see condensation of chromosomes. Okay, condensation of chromosomes, spindle fibers, centrioles at opposite poles. These are the main characteristics of this stage. Okay, now let's move to metaphase. The completion of prophase is characterized by, okay, what are the main characteristics of this phase? We will be discussing that. The microtubules start pulling the chromosome with equal force, and the chromosome end up in the middle of the cell. See, they will end up in the middle of the cell, like over here. Right now, they are not in the middle of the cell. Okay, haphazardly arranged. In metaphase, they get pulled by the spindle fibers and then they lie on the equatorial plane. Equatorial plane is the keyword over here. equatorial plane or the metaphase plate. Okay, yeah, they come and lie at the center of the cell. Okay, like this. You can see this is very important. This is one of the characteristics of metaphase. Okay, ends up in the middle of the cell. This region is known as metaphase plate. Like I told you, this is also known as equatorial plane. You must have seen the Earth. We have an equator, which is the middle line passing through the Earth. That is the equator. So that is the reason it is also known as equatorial plane. Thus, each cell gets an entire functioning genome. Okay, so these chromosomes, ends of the chromosome are pulled towards the opposite plane, and in the next phase they will divide. Okay, so that is the reason it is said each cell gets an entire functioning chromosome. Now let's move to some question answers. You will know. Okay, this stage in which chromosome align on the equator of spindle fiber is, like I told you, metaphase. Okay, now you know what kind of questions might appear in your exam. So the stage in which chromosome align on the equator of spindle fiber. Okay, or equator of the cell. Okay, equatorial plane or metaphase plate. Obviously, they will not give you metaphase plate because it will be very obvious. The answer will be very obvious. Mainly, it is equatorial plane or equator of the spindle fiber. Okay. And the next question is the best stage at which the total number of chromosomes can be counted in any species is metaphase. Yes, this is a very prominent stage to study any cell division. Okay, during metaphase, if you look under the microscope, metaphase is clearly visible. It is the most clear stage that is visible under the microscope. Okay, metaphase will be very clear because the chromosomes are placed on the equatorial plane and they're very, very visible. It is very, you can just look at it and know, yeah, this is metaphase. Okay, so one more thing. Okay, one more thing I want to tell you. During prophase, what happens? All the other, all the other, what should I say, cell organelles start to disappear. You will not be seeing them. Okay, during, if you see it under a light microscope, okay, you will not be seeing it because whatever we are discussing under what has been seen under the light microscope. So these are the principal properties that are seen of each stage. Okay? So let's come to anaphase. The splitting of sister chromatids mark the onset of anaphase. So the chromatids split. Okay, they are split towards the opposite poles and move towards the opposite poles. Okay, these sister chromatids become the chromosome of the daughter nuclei. Okay, the chromosome are then pulled towards the pole by the fiber attached to the kinetochore of each chromosome. Okay, and then the centromere of each chromosome leads at the edge while the arm trails behind it. So it will always happen that the centromere will be the one leading towards the centriole. It will not be the arms. Okay, these arms will not be leading to the centriole. Okay, so the arms are the tail part and centriole is the head part while moving towards the opposite pole during anaphase. Okay, it is one of the major characteristics that the chromosomes divide. Okay, chromosome divide and move to the opposite poles during anaphase. Okay. Now let's move to question. The characteristic of this stage of mitosis is the separation of sister chromatids. Very obvious, it is anaphase. Like we discussed, this is when one of the prominent characteristics that the sister chromatids separate and move to the opposite poles and are led by the centromere, and the arms are the tail part, which trail behind it. Okay, let's move to telophase now. The chromosome that cluster at the chromosome that cluster at the two poles starts to unite into undifferentiated mass, as you can see. Okay, as the nuclear envelope starts to form around. Okay, so the nuclear envelope also forms around it. Earlier, it was disintegrated, it disappeared. Okay, now it is reappearing. All the cell organelles will start reappearing in the stage. Okay, so Golgi bodies, ER complex, which had disappeared after prophase, start to reappear. A reappearance of an enlargement of the nucleus. Okay, nucleus will also form. Okay, you will see a nucleus because of the presence of nuclear membrane. It is dividing the cytoplasm and the nucleus content. Okay, enlargement of the daughter nuclei. Okay, to that interphase, decondensation of chromatin. The chromosomes will decondense. Okay, okay. The characteristic of this stage, decondensation of chromatids. What is it? Telophase. Okay. When after the whole cycle, mitosis, when everything starts to reappear, all the cell organelles and the nuclear membrane and the nuclear contents have reached towards the opposite side. One centrioles are also present. So are you seeing both have equal amount of content in them? So these will only divide into the two daughter cells. Okay. Now we move to cytokinesis. In this phase, the cytoplasm of the cell divides. It begins as soon as the mitosis ends. Okay, after mitosis, cytokinesis happens. So after this stage, the cytoplasm will also divide. This, do you see this cleavage? This will end. This will turn into, I would say this will diminish. Okay, and these two daughter cells will be separated. Okay. Plant cells are much tougher than animal cells as they have a rigid cell wall and high internal pressure. The cytokinesis occurs in plant and animal cells differently. So let me tell you how it happens in plant cells. In plant cells, plate formation takes place, and in animal cells, cleavage formation takes place. Okay. This is a cleavage. You can see a cleavage. And in case of plant cell, they divide by a plate system. Like, let me share. So let's get rid of this. So let's say this is a plant cell dividing. Okay. And this is the plate formation that will happen. This is the nuclear material. Sorry, the nucleus becomes really big. Okay. And in case of animal cells, it happens somewhat like this. There is a cleavage formation like this. Okay. This is the cleavage. And this is the plate in case of plant cells. Okay, got it? So, where is that? Okay, let's do a quick question on cytokinesis. Okay. The division of cytoplasm is known as cytokinesis. Okay. So basically, what is cytokinesis? Division of cytoplasm. And okay, so let's do a quick revision of whatever we have learned today. We have learned what is cell division. Okay. Basically, what is cell cycle? So cell cycle is a series of events, okay, that takes place during cell division. Okay. It is marked by duplication of DNA, division of cytoplasm and organelles, and production of two daughter cells. Okay. Identical daughter cells. They will be identical in structure. Okay. Okay. So what is the importance of cell division? So now that we have learned about mitosis, let's see what is the importance. Okay, why is it necessary? So cell division is important because of the following reasons. It helps in survival and growth. So in order to grow, the cell needs to divide. Unless and until it will not divide, how will there be many more cells? Okay. So in order to survive, to repair itself from the damages, it goes under cell cycle. Okay? Maintenance of the chromosome number, like I told you. Okay, to maintain the chromosome number as well, there are certain kind of cell division that takes place. Okay, so what is the cell cycle? Just told you about the discoverers Prevost Dumas in 1824. Okay, okay. So this is the entire process. It is basically series of events by which from one parent cell, new cell population grows. Okay. Then we come to types of division. We learned about mitosis, meiosis, binary fission. Then phases of cell cycle. There are two major phases, interphase and mphase. And under interphase, there is G0 phase, G1, S, and S2 phase. G2 phase. Then we did question answers. Then we did M phase. M phase is basically mitosis or equational division. Then we did prophase. Prophase main characteristic is disintegration of the nuclear envelope, decondensation of what? The chromosomes. Okay. So, and the centrioles move to opposite poles. Now we come to metaphase. Metaphase, the chromosome line on the equatorial plane. Anaphase, the chromosome move to the opposite poles. Okay. And the chromosomes also divide. Okay. These are sister chromatids. Okay. These two hands, these are sister chromatids. So they divide and move to the opposite poles in anaphase. Okay. And in telophase, nuclear membrane reappears, all the cell organelles reappear, the spindle fibers disintegrate. Okay, there are no more spindle fibers in this stage. Okay, each cell will have their own centrioles, two centrioles at the end, but no spindle fibers over here now. Okay, no spindle fibers, only cleavage. Okay, so and there is cleavage, and in case of plant cells, there is a plate formation. Then cytokinesis. Cytokinesis is the division of cytoplasm. Okay, so this is it for today. Thank you.